Hello, here I will talk about the use of real-time genomics for fast and portable avian influenza sequencing. I will talk about the avian influenza as a health disease and how we use real-time genomic for rapid genetic characterization of the avian influenza virus for mammalian adaptive variants detection and also for environmental surveillance. First, I would like to talk you about the avian influenza disease, that it's affecting a bunch of species, as you know, for the last H5 hypopathogenic avian influenza virus strains that are causing huge mortalities in wild birds and also wild mammals. But also, from the domestic species, that is also causing huge mortalities in poultry, but also in domestic mammals, as minks, cats, and cows recently in the United States. Also, sporadically, there is a few cases in humans, but luckily not yet with human-to-human transmission. This disease is caused by an RNA virus that is in constant evolution due to the error-prone polymerase. And this justifies the use of genomics for understanding this evolution. Now, I would like to show you how we use real-time genomics for rapid genetic characterization of this virus. What we did was to use an isolated virus in a specific pathogen-free X, and therefore it was a high concentrated virus. And then we performed RNA extraction and the quantification with quantitative PCR. Then we performed two different kinds of strategies for sequencing. First, the direct RNA sequencing with the sequence-specific protocol that allowed us to only sequence the viral RNA from influenza virus. The other strategy was to perform the rapid barcoding kit after the conversion to the viral RNA to cDNA and amplification of universal primers for avian influenza. It's important to mention here that we use the latest chemistry from Oxford Nanopore in both cases. For the bioinformatic analysis, what we did was first the base calling, then selection of the best reference from a database from NCBI that had all the influenza strains, and then with the best reference we built a consensus and then a phylogenetic tree. These are the first results of this work, where we show that both approaches work really good, but there is a slightly better coverage, better read and consensus level accuracy with the DNA approach. That's why we still suggest that this is better for low concentrated samples to use this kind of approach. Also, it offers an easier protocol with more throughput, better accuracy, and also the multiplexing option. That this means to use more than one sample in the same sequencing run. However, there is also the option to use direct RNA sequencing for some specific scientific questions, because you don't have to do amplification or conversion to cDNA. And this allows you to detect RNA modifications that are known to play a role in the evasion of the innate immune system in the host. To validate these results, we use actual environmental samples from a farm in France, a dog farm in France that we know that was infected with a high pathogenic H5N1 strain. We use those samples, and for that we sequence using the cDNA conversion and amplification, because we determined previously that the limit of detection of the direct RNA technique was too low for these environmental samples. So after performing this protocol, we were able to obtain the consensus from all the segments and build a phylogenetic tree showing that they were clustering together with other H5N1 strain, as we expected, because we knew that was the virus that was infecting the farm, and therefore validating our protocol. If you want to know more, we recently published these results in Virus Evolution. Now, I would like to show you how we use these approaches to detect mammal-adapted variants. For that, we used an in vivo experiment in a BSL-3 facility in Barcelona, where we inoculated a group of minks with a H5N1 high pathogenic strain that was adapted to wild birds. what we did was to infect a group of minks to understand how this wild bird adapted virus was adapting to mammals. What we saw is that at the end of the experiment, at 16 days post-challenge, there were 2 animals that were positive in brain. And what we did is collect these samples and sequence together with the original inoculum to compare the different variants. What we did is perform RNA extraction, the amplification of all the segments of the virus with these universal primers I told you before, and the rapid barcoding kit sequencing. It's important also to mention here that this is a good example of the portability of this technology because it allows us to perform this inside the BSL tree, reducing the biosafety risk of this method. Then, for the bioinformatic analysis, what we did it was the super accuracy base calling, then the mapping with Minimap2, then we use Clear3 for variant calling, and SNPF annotation tool for the annotation of the variants, and finally the classification of these variants with the website tool Flusurber. It was interesting in the Mink4 to observe that there were a few of spontaneous mutations occurring that could be related to mammal adaptation. But specifically, we focus attention in one, in the polymerase base 2 at the position 271, because it's a well-known mammal adaptation variant that indeed it was present in all the minks in an outbreak in a farmed mink in Spain. Also, it is known that this variant enhanced the polymerase activity in mammalian host cells and mice, and it was also previously found in the pandemic H1N1 strain. This is, to the best of our knowledge, the first reported observation of this spontaneous occurrence of this variant in a mammal. Also, I would like to show you how we can use these approaches for environmental surveillance. For that, we take advantage of a pilot project grant from the German One Health platform that allow us to collect samples from different wetlands where we selected depending of if they had anthropogenic pressure or not anthropogenic pressure. So in each of these wetlands, we collected different environmental samples, and in each of these samples, we try to detect the avian influenza virus by quantitative PCR, but also sequencing the positive samples. Also, we perform a general virome analysis and also the detection of antimicrobial resistant genes in the bacteria microorganism. However, I will not have time to explain this last part of the project. These are the samples that the samplers that we use to collect environmental samples, to collect air and water, but I want to pay specific attention to these torpedo devices, torpedoes-like devices that have electronegative membranes inside and that collect water in a passive way, because this is the samples that we processed first and that I will show you next the first results. So with these devices, we were able to detect influenza from all the wetlands in the French locations that were close to duck farms. However, the virus detected there were in low viral concentration and with the fragmented RNA, what hampered our amplification with the universal primers that I explained you before. However, we were able to sequence the hemagglutinin segment from one of the samples, and this showed us that was clustering very close to an H4, so a low pathogenic strain from duck origin. What have sense because, as I explained you before, these wetlands were close to duck farms. Now, moving to the more general virome analysis, what we did was a random amplification using SMART9N. That's a similar technique than CISPAT that you are probably aware of that, but it's adapted for long reads. Here, what we saw is that the main hits were related to plant, prokaryote, or insect virus, as we expected, because these were environmental samples. But the host, the families that we know that have animal as a host, that was Picornaviridae and Nodaviridae, were mainly detected in the anthropogenic environments, as you can see in the figure below. So that's all from my side. I would like to thank everyone for the help with this work, especially my PI, Dr. Lara Urban, and thank you everyone for listening to me.